Hey guys, so we're here for non-Mendelian genetics 1. Okay. So we've been looking at the Mendelian genetics where basically we have um, dominant and recessive, but we found that many of the traits don't actually follow the ratios that were predicted by Mendel's laws. And so the question comes up, well, why don't they fit? He did all that research and this is what he found. The reality is he just looked at a small portion of everything that makes up genetics. So now we want to think about the fact that there are various degrees of dominance in genetics. Sorry. Many traits are produced through multiple genes. So the reality is a lot of our different features that we think about, hair color, eye color, some of these things where there's a lot of variation, the reality is there's more than one gene involved in determining those factors. Additionally, some traits are determined by genes that are on the sex chromosome. A Y related gene is only going to be found on the Y chromosome. Only males will ever have that. An X-linked gene might show up for females or it might not. It depends on if they have two copies of it, one copy of it. We don't know because it depends on those sex chromosomes that they end up with. And then we've also found that sometimes there are genes that are really close to each other or right next to each other on the same chromosome. And so the reality, we talked about how that whole, they'll separate out and they won't get sorted together. But what we realize now that's a little different from what Mendel found was that if they're close enough on the same chromosome, they're going to separate out, they're going to be transferred together. And so typically that's going to limit our diversity a little bit because they tend to move together. And then, of course, there are also a few traits that are a result of what we call non-nuclear inheritance. What this is really trying to say to us is that they are typically found on something like the chloroplast or the mitochondrial DNA, which is located in those organelles, not part of the main nuclear DNA. So we'll start with degrees of dominance. So like I said, alleles can show varying degrees of dominance. In Mendel's experiment, he worked with traits that were completely dominant. So if that dominant was there, that's all we ever saw. Homozygous dominant, heterozygous individuals, phenotypically, they appeared the same. But we have another possibility, which is called incomplete dominance. And the idea here is neither of them is really fully dominant. And so what happens is in that F1 generation, in that mixed heterozygous generation, we kind of blend together to form something new. So think of it kind of like blending paints. If you take yellow paint and blue paint and blend them together, you get green. Here we get an example. Red flowers crossed with white flowers produce pink. Do the same thing with paint, right? So the idea is that neither of them is completely dominant. So when we mix the two together, they blend into kind of a third option. Then we have codominance. So we're saying that both of these are equal to each other. And so you'll see both of them present if they're both present. So one of the most common examples tied to codominance has to do with blood typing, and specifically with type AB blood, where we see both the A and the B present. So A and B are codominant to each other, hence they make type AB blood. Then there is that concept of multiple alleles. So the genes have forms that have more than 2 alleles. So once again, human blood group actually falls into this because we know that A and B are codominant to each other. But if you think about it, when we talk about blood types, we have B, AB, and O. So we have IA, IB, or a lowercase I is what represents O. Sometimes you'll see it just with the letters, but this is actually the formal official format. And so we've got multiple situations going on where we have dominant and recessive and codominant because we have more than 2 alleles that mix together to make our system. Okay, so we have a black mouse crossed with a white mouse. The resulting offspring are gray. What's the easiest explanation? And so this is one of those, think about the paint. We mix the 2 together and we get a third option. So if we have a heterozygous mouse here, our black mouse, capital B, and we get a lowercase b from the white mouse, when we have the capital B, lowercase b, we end up with a gray mouse. So it's incomplete dominance. Okay, cattle can be red, all red hair, white, all white hair, or roan, red and white hair. We're seeing both. What's the best explanation for this phenomena? So if we're seeing both of them equally, then that would be codominance. A red cow is crossed with a roan cow, and roan is that red and white. What would the phenotypic ratio of the offspring be? So we're going to cross red with RW roan. We do the Punnett square. We have a 1-to-1 ratio, 50% are red, 50% are roan. Okay, a woman with type A blood has a child with a man who has type B blood. With this limited information, what are possible genotypes for the woman, for the man? So if we have type A blood, well, obviously we can have AA because that's all we get. But remember how we talked about this situation? What about O? And we realized that O is recessive to A and B. So we could have AO. Same thing with the male for B. He can be BB or BO. Okay, and then we have the multiple genes. So in a lot of cases, we have 2 or more genes responsible for determining phenotypes. So here you'll notice we have a series of mice. And it looks like we have kind of beigey, they're called agouti, that beigey brown color. We have black mice and we have albino or white mice. So epistasis is the phenotypic expression of a gene at one locus affects a gene at another. So the idea here is that if we have one specific gene, it's going to then affect the results we see from another gene. So there's one gene that codes for pigment. And then there's a second gene that determines whether or not pigment will actually be deposited. So we have the gene that codes for, are they agouti or are they black? But then there's another gene that says, do we even have pigment or is there no pigment? And so when we get the gene that says no pigment, we end up with those albino mice, even though they may have the other gene that says they should be agouti. So both genes have an effect on what we see overall. And then we have polygenic inheritance. So the effect of 2 or more genes acting on a single phenotype. And so the idea here is you can see in this picture, we have 3 different genes. And based on how these different ones come together, give us these different color palettes. So height, human skin, things like that that have so much variation, we're going to put together multiple, and based on the combination determines our skin coloration, or might determine how tall or short we are. Okay, so that is it for this non-Mendelian 1. We'll be back in our next lesson with non-Mendelian 2.